Hello everyone. Earlier in our classroom discussion, we have discussed about glycolysis, TCA cycle or Krebs cycle, and those cycles were primarily concerned with the generation of ATP, starting with glucose as fuel. We now turn to the generation of different types of metabolic energy, that is reducing power. Many cells possess another pathway of glucose degradation known as the pentose phosphate pathway or phosphogluconate pathway or hexose monophosphate pathway. This is not the main pathway for obtaining energy from the oxidation of glucose in animal tissues. Instead, it is a multifunctional pathway specialized to carry out four main activities depending on the organism and its metabolic state. Its primary purpose in most cells to generate reducing power in the form of NADPH. This function is especially prominent in tissues like the liver, mammary gland, adrenal cortex, and the adipose tissues that actively carry out the reductive synthesis of fatty acids and steroids from acetyl-CoA. Skeletal muscle, which is not active in synthesizing fatty acids, virtually lacks this pathway. The second special function of this pathway is to convert hexoses into pentoses, particularly D-ribose 5-phosphate. These pentose sugars are required in the synthesis of nucleotides as well as nucleic acids, that is ribonucleic acid RNA, deoxyribonucleic acid DNA. The third function is the complete oxidative degradation of pentose sugars by converting them into hexoses, which can then enter into the glycolytic sequence. In the fourth function, pentose phosphate pathway can be modified so as to participate in the formation of glucose from carbon dioxide in the dark reactions of photosynthesis in case of plant. The reactions of the pentose phosphate pathway takes place in the cytoplasm of animal cells. The pathway can be divided into two branches, oxidative branch and non-oxidative branch. In oxidative branch, the reactions which occur involve oxidation reduction reactions. The oxidative branch of the pentose phosphate pathway starts with the dehydrogenation of glucose 6-phosphate at carbon number 1. The reaction is catalyzed by the enzyme glucose 6-phosphate dehydrogenase, and it is highly specific for NADP+. That is the nucleotide adenine dinucleotide phosphate. The product of this reaction is 6-phosphoglucono-delta-lactone and NADPH, and the reaction is irreversible, that is occurs only in forward direction. The next step is the hydrolysis of 6-phosphoglucono-delta-lactone by a specific lactonase enzyme to give 6-phosphogluconate. Here is the enzyme is 6-phosphogluconolactonase. In the next step, 6-phosphogluconate undergoes oxidation and decarboxylation by 6-phosphogluconate dehydrogenase to form ribulose 5-phosphate and NADPH. 6-phosphogluconate dehydrogenase is a magnesium dependent enzyme. Now the final step in the oxidative branch of reactions is the synthesis of ribose 5-phosphate by the isomerization of ribulose 5-phosphate, and the responsible enzyme is phosphopentose isomerase, previously known as ribose 5-phosphate isomerase. This is an isomerization reaction. Now, under some metabolic circumstances, the pentose phosphate pathway ends at this point, and its overall equation may be written as following: glucose 6-phosphate plus 2 NADP plus H2O to ribose 5-phosphate plus NADPH plus carbon dioxide plus 2 hydrogen ion. So the net result is the production of NADPH for reductive biosynthetic reactions in the cytosol, and the production of ribose 5-phosphate as a precursor of nucleotide synthesis. However, many cells need NADPH for reductive biosynthesis much more than the ribose 5-phosphate for incorporation into nucleotides and nucleic acids. In these cases, ribose 5-phosphate is converted into glyceraldehyde 3-phosphate and fructose 6-phosphate by transketolase and transandrolase enzymes. These enzymes create a reversible link between the pentose phosphate pathway and glycolysis by catalyzing the reactions from two C5 compounds to C3 and C7. After that, from C3 and C7 can produce C4 and C6 compounds, and after that C4 and C5 compounds produce C3 and C6, that is the glyceraldehyde 3-phosphate and fructose 6-phosphate. So the first of the three reactions linking the pentose phosphate pathway and glycolysis is the formation of glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate from two pentose sugar, two C5 compounds, ribose 5-phosphate and xylulose 5-phosphate. This reaction is catalyzed by transketolase, which contains tightly bound thiamine pyrophosphate. Thiamine pyrophosphate is a derivative of vitamin B1, thiamine. These transketolase enzymes also require magnesium ion. Now, xylulose 5-phosphate is produced from ribulose 5-phosphate through the epimerization by the enzyme phosphopentose epimerase. We will discuss about this in later also. Now, glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate react to form fructose 6-phosphate and erythrose 4-phosphate. This reaction is catalyzed by transaldolase. In the third reaction, transketolase catalyzes the synthesis of fructose 6-phosphate and glyceraldehyde 3-phosphate from erythrose 4-phosphate and xylulose 5-phosphate. So, The sum of these reactions is 2 xylulose 5-phosphate plus ribose 5-phosphate will produce 2 fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. Xylulose 5-phosphate can be formed from ribose 5-phosphate by the sequential reaction of pentose, phosphopentose isomerase and phosphopentose epimerase. And so the net reaction starting from the ribose 5-phosphate is according to the follows. 3 ribose 5-phosphate will ultimately produce 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. Thus, excess ribose 5-phosphate formed by the pentose phosphate pathway can be completely converted into glycolytic intermediates. So, it is very much evident that the carbon skeletons of sugars can be extensively rearranged to meet the physiologic needs. That's all. Thank you.